For those of you who don't know me, I'm Laurel Buxbaum, and I want to welcome everyone to the Shrier Family Topics in Rehabilitation Science lecture series. And I'm really delighted to introduce my friend and colleague, Dr. Roy Hamilton. I was trying to think, Roy, of how long we've known each other, and I think it's been, I don't know, more than 20 years,
Decades,
probably.
yes, in the
Yeah,
multiple decades at this point, yeah.
and definitely in the double digits. And Roy is an associate professor of neurology and PM&R at the University of Pennsylvania, where he directs the lab for cognition and neural stimulation, the LCNS lab. And he's quite well known, deservedly so, for his work using transcranial magnetic stimulation and TDCS, transcranial direct current stimulation. Roy is also the director of the Penn Clinical Neuroscience Training Program. He's the assistant dean of diversity and inclusion for the Perelman School of Medicine and the vice chair of diversity and inclusion for the Department of Neurology at Penn. And Roy has won numerous awards, including the American Academy of Neurology's Norman Geschwind Award. And he's a prolific guy, a great person to work with and collaborate with. And I just want to extend the warmest of welcomes to you. Thanks, Roy.
Thank you so much, Laurel, for that warm introduction. It's great talking to this crowd. It's always nice getting together with old friends, and also, those of you who I don't know, to talk about the work that we've been doing. I would like to describe what I'm going to talk about in the next hour as being one part historical, one part apocryphal, and one part aspirational. And what I mean by that is I'm going to go through the history of the work that we've been doing in non-invasive brain stimulation. So that's the historical part. I'm going to frame it around a debate about a pretty simple model about how neuromodulation in the language system works. Probably too simple to be the end all be all. So that's the apocryphal part. And then at the end, we'll spend some time talking about what I think is the future, or at least our future in the lab with respect to this kind of work. So that's the aspirational portion. So let's get through some obligatory slides first. So the lecture is supported by a generous gift from Nancy and Marc Shrier. So thank you for the gift. It doesn't seem like anyone has any irrelevant financial relationships disclosed for this lecture, myself included. And these are the learning objectives. Don't bother reading these in detail because I'm going to summarize them in the next slide, which is, this is our roadmap for today. So one topic that I'm going to talk about in some degree of detail is the idea of interhemispheric interactions in post-stroke aphasia. And one reason why I'm going to talk about this notion is that it is one that has animated the field of non-invasive brain stimulation as it pertains to aphasia recovery for a number of years. So while those of you who are aphasiologists or interested in the language system may not think as hard about those interactions, it turns out that they've been very influential in how people who do neuromodulation think about them. So I'm going to sort of set up the idea as to why that's the case, and then talk about a little bit about how I criticize, I think, some of the ideas and some of the simplistic models that people build around that idea. Then we'll talk about transcranial magnetic stimulation. It's used for post-stroke aphasia. I'll introduce the idea of transcranial direct current stimulation, the other workhorse in laboratory for post-stroke aphasia and for neurodegenerative aphasias. And then we'll finish by taking a look at challenges and next steps for neuromodulation in the field of aphasia. So I feel like starting out with the impact slide for this audience, a little bit like bringing coals to Newcastle, but I'm going to do it anyway. So stroke is a highly prevalent disorder. The lifetime incidence of stroke globally is about 1 in 6, which is very high. Aphasia, the acquired loss of language ability, is the most common focal cognitive deficit associated with stroke, depending on which review you read. It might range between the 20% to 40%. So let's split the difference and call it a third. That's still an enormous number of individuals who at some point in the course of their post-stroke recovery will experience problems with communication on the basis of having had a stroke. And while we do have behavioral interventions that have some efficacy, we don't yet have targeted neurologic interventions or based on that address the specific neural basis of aphasia and aphasia recovery using sort of guided technologies. And that's really what we've been trying to do with non-invasive neuromodulation for the last number of years. So if we're going to apply guided technologies to specific parts of the brain in order to try and address aphasia and aphasia recovery, we have to have some understanding of what areas of the brain are relevant in individuals who have aphasia and who have communication deficits on that basis over the course of their recovery and thereafter. So I'm going to start here. This is work that we did a number of years ago. This is with Peter Turkeltaub. I'm sure a number of you know who that is. He was a fellow at the University of Pennsylvania in behavioral neurology. This is work that he had done way back then, actually before the year listed here at the bottom of the slide. And now is a behavioral neurologist, a physiologist in his own right at Georgetown University. And what we're showing in this slide here is the difference in the constellation of brain areas that are engaged during the same sets of language tasks in individuals with aphasia, who are depicted on the bottom, compared to individuals who are healthy language users. And what you can see, completely aligned with common sense intuitions from clinical and behavioral neurology, that individuals who are healthy language users, they activate a left hemisphere network, a largely left hemisphere network of areas, the geography of which should look pretty familiar to people who are familiar with this literature. If you compare that to individuals who have aphasia, and in this case, the figure is showing persons with chronic aphasia, what you see is something different. You see a bilateral constellation of brain areas that appear to be activated during these same tasks that activate a left hemisphere dominant constellation of regions in controls. Right? And so we're going to talk a little bit about what could be going on in that right hemisphere, right, when those brain areas are active. And again, we're going to sort of harkening back to what I was saying about this being a little apocryphal, we're going to sort of divide this up into some pretty basic ideas, some basic categories of what could be happening in the right hemisphere of individuals with chronic aphasia. So let's start with this almost silly, for its simplicity, cartoon here of how an intact system might be working, where what I'm showing in the green is a completely topographically not representative sort of model of, sort of an active regions of the left hemisphere in individuals who are healthy language users, right? So, in this cartoon, if you have a lesion of the left hemisphere that in an area that's important for language, or the network of areas that are important for language, one thing that might occur is that regions that are close to, adjacent to, or we're part of that language network, so what we call perilesional areas, may be recruited, may be engaged, and that engagement may be compensatory in nature. That is something that, at least in the field of neuromodulation for aphasia, there's relative consensus on. So we're not going to actually spend a ton of time talking about that. We're going to spend more time talking about three basic ideas of what could be going on the right. So one idea is that engagement of right hemisphere areas is compensatory too, right? It's you're missing regions of the left hemisphere, and so the right hemisphere now, parts of it come online as a compensatory strategy, if you will, for recovering language abilities. Another, and in the TMS literature, let me say a very popular idea, is the idea of interhemispheric inhibition as the mode of interaction. And what that means is that, we'll get into it in a second, but that the activity of the right hemisphere is not helpful, but that it in fact is deleterious. And I'll go over a couple slides explaining how that model works. And then finally, this is just a sort of cartoonish representation of the idea that the model might not be so simple, right? That there might be compensatory activity of the right hemisphere to some extent, but that compensation might be inefficient, that there might be areas that are useful, other areas that are deleterious, so sort of a mix, and that you might be able to do something to increase the efficiency of a sort of inefficient compensatory system. Right, so let's take some bits of evidence. For the purposes of time, we don't have time to go over all the evidence of all of these last three theories of what the right hemisphere might be doing. So I'm just going to provide you a couple of snapshots. For my snapshot of compensatory activity in the right hemisphere, I'm going to go all the way back to 1877. Although I could point to later bits of evidence, I like the historicity of this. So in 1877, a physician named Thomas Barlow was treating a 10-year-old child who unfortunately had infective endocarditis. In other words, an infection in their heart that was unfortunately throwing emboli to various parts of the vascular system, right? And so in the context of that 10-year-old suffered a stroke. And here is how Barlow describes that child's abilities shortly after they suffer from their first from their stroke. He had lost his speech. He could only say ha ha. He could not grasp with the right hand, and he dragged the right leg when put on his feet. Now, as is often the case in children who have brain injuries, this child made a pretty remarkable recovery. Here's how Barlow describes him 10 days later. In 10 days, he was greatly improved. The speech had returned on the 10th day, but occasionally he made a mistake, gave the wrong name for a boy, and did not seem always quite to understand what was said to him. In a month's time, he seemed quite to have recovered. All right, that's great. So this child, you might imagine, had suffered from a left hemisphere stroke and had some impairment of his language ability. A hint to the fact that it's in the left hemisphere is his right hemiparesis, sort of at the top of the paragraph. But unfortunately, one thing that they didn't have in 1877 is good cures for infective endocarditis. So unfortunately, this child had a second stroke. And when the child had a second stroke, here's what Barlow had to say about his deficits. The only approach he could make to a voluntary articulate sound was ah. He could cry vigorously. There was no lack of voice. From his admission, he appeared to understand all that was said to him. When asked his age, he counted 10 on the questioner's fingers, counted 4 when asked how many brothers he had, and so forth. He made signs for what he wanted, and the signs were correct. Basically, Barlow is saying that he had recurrence of his aphasia. And unfortunately, in the absence of cures for the underlying disease, this child passed away. And when he went to autopsy, what Barlow noted was indeed, he had had an initial stroke of the left hemisphere, causing him to have his initial deficits, but he also had a second stroke of the right hemisphere in areas that were homologues, sort of mirror images of areas that in the left hemisphere we would typically associate with language. Right? And so the implication here, and it's been shown in other cases as well, and in some imaging, but the implication is that at least in this case, the right hemisphere was important for mediating this child's recovery from aphasia initially, as evidenced by the fact that a second lesion to that right hemisphere was so deleterious. Right? So now let's shift gears to that idea that the right hemisphere might be harmful. Where does that idea come from? That idea is born of the notion of interhemispheric inhibition, a very popular idea in the field of neuromodulation. Here's how that model works. It's predicated on the idea that the two hemispheres, while richly connected through the corpus callosum, that many of those connections are actually inhibitory in nature, such that if you have a sizable lesion on one hemisphere, one of the downstream effects of that would be to release the other hemisphere from inhibition, that it would become increasingly active. However, that increased activity wouldn't be representative necessarily of a compensatory response. It would just be a release from inhibition. What's worse, according to this model, that increased activity could itself then project inhibitory transcallosal connections back to the already damaged, already hampered, if you will, hemisphere, and further inhibit recovery in, say, for example, those perilesional areas that we discussed early on of the left hemisphere that we agreed might be compensatory in nature in individuals with stroke and aphasia. So that's how this model works. And you know, it's not drawn out of thin air. I mean, it's the kind of thing that you can demonstrate to some extent in the motor system with individuals with stroke and paresis. So, for instance, you can take individuals who've had unilateral strokes in the motor system, and obviously, if you try to elicit a response, a motor response, let's say you put a transcranial magnetic stimulation coil over that part of their head, their brain, and try to elicit a motor response, you'll obviously elicit a weaker response over the lesion hemisphere. But what might have previously surprised you would be that you also elicit a stronger response from the intact hemisphere, right? The intact hemisphere's motor response is actually facilitated because it's been released from inhibition, right? And you can actually equilibrate those motor responses one of two ways. You can try to inhibit it, well, you could try to facilitate the damaged side, right? That's intuitive. But in line with this interhemispheric inhibition idea, you can also try to inhibit the intact side as a way of equilibrating those motor evoked potentials, right? And so there's some purchase there with respect to the idea that these inhibitory interactions are relevant. And a number of years ago, people would make this argument with respect to elements of the language system. So here, for example, is a pretty old article now, but from a number of years ago, in which the investigators contrasted brain activity when individuals were performing a naming task. And what I'm showing you here is just subtractions when they got the correct versus incorrect response. And so just to point to the bottom line, literally, is that when individuals were naming incorrectly in this particular study, they were more likely to engage their right hemisphere. And so by this line of logic, this author and others sort of put forth the argument that, well, maybe the activity of the right hemisphere isn't actually beneficial. Maybe it's either spurious or, worst case scenario, adding unhelpful noise to the brain's response. All right, so these are a couple of the ideas that when we started our work, we were interested in disambiguating. And so going back to that work from Peter from a number of years ago, we identified this constellation of areas, including right hemisphere areas in patients with chronic stroke and aphasia that seem to be active during language tasks. Great, but that doesn't address whether those areas are helpful or not. And so as first pass, we went through a pretty simple algorithmic analysis. What we did was we looked at each of those spots in the brain and we said, look, for each of those areas, we asked two questions. Is that area a homotope? In other words, is it sort of a mirror image area of a region on the left hemisphere that normally is engaged in language tasks for healthy individuals? Also, is it a homologue? In other words, if you look across the various tasks where a particular area is activated in the left hemisphere, if you look at that region in the right hemisphere, is that area activated for that same pattern of tasks? So yes, for task 1 and task 2, but not for task 3. No for task 4 and 5. Yes for task 6, right? Does it have the same patterns of activation? Because if the answer is yes, it seems to have the same patterns of activation as a homotope on the other side, well, that might give you some hint that it's at least somehow relevant to the performance of those sets of tasks. Whereas if it was completely orthogonal, just random noise compared to what was going on in the left hemisphere of healthy individuals, that might suggest to you that it's not contributing in any meaningful way to the task. And to make a long story short, we chose these areas to modulate with TMS as a way to try and figure out, in addition to some of the imaging work that was done, whether or not these areas were functionally going to be relevant. And I'll show you a little bit of that data too. Now, before I move on. So one thing that we identified was that in the functional imaging analysis, there was this one area shown here, area H, the pars triangularis on the right. Remember, I was dividing up these brain areas into homotopes and homologues. Well, it's certainly homotopic to an area in the left hemisphere that's relevant, that is Broca's area, but it seemed to be not homologous in its patterns of activity. It seemed to be one area that seemed to be most orthogonal with respect to its patterns of activation across these various tasks. So we'll revisit that later. So now let's talk a little bit about the idea of trying to interrogate these various areas with brain stimulation. At this point now, 10, 15 years on, I no longer have to give a sort of lengthy explanation of what TMS is to most audiences. So, but I'll give this snapshot, right? So transcranial magnetic stimulation involves applying a coil of wire, encased usually in plastic, a coil of wire, you place it adjacent to the scalp. You're then going to discharge a huge current through it. Okay. At the moment that you discharge that, it creates a fluxing magnetic field. That fluxing magnetic field is sufficient in its intensity to then depolarize underlying cortical neurons in a focal manner. Right. And so that that allows you to modulate brain activity in a relatively high resolution way. It's about, think of it as being about the area, a centimeter and a half. So about the area of your thumbnail, sort of searching over the surface of your cortex. Depending on how you apply it, it can be done in an inhibitory or excitatory manner. That depends on the pulses and the patterns of pulses that you give. It's safe as long as you're giving it within accepted safety parameters. The one thing you're trying to avoid is having a seizure, but again, as long as you're within accepted safety parameters, that generally doesn't happen. And if you apply it repeatedly, you can induce long-term changes in cortical activity that we think are related to long-term potentiation-like effects. All right, so in some of our early work, what we did was we brought individuals in and we started searching around in various areas of the right hemisphere, right? And a phase that we called site finding. And in this case, we were using inhibitory stimulation. So 1 hertz, I'm not going to go into the mechanics of how different frequencies give different effects. Let's just say 1 hertz is inhibitory stimulation. So we're giving inhibitory stimulation over the right hemisphere, informed largely, I must say, by that interhemispheric inhibition model that I was telling you about a few minutes ago. So we're searching around in that right hemisphere for an area where inhibiting it results in some at least temporary benefit in language performance. And if we found the spot, well, what we did was we enrolled these individuals in a sham-controlled TMS trial in which they either received 10 days of TMS to that best spot that we stimulated, or sham stimulation to that best spot. We followed them up for a couple of months. We didn't have the heart to keep all the people who were getting fake stimulation in the fake stimulation arm for the entire 6 months of study. So there was a partial crossover. The people in 2 months crossed over so that everyone ended up getting stimulation, and then followed everyone out for 6 months. And what we found in this pretty early work was that indeed, after 10 sessions of 1 hertz inhibitory stimulation, often to the right inferior frontal gyrus, we'll get to that in a second, individuals had improvements in different aspects of language performance. For example, this is a graph showing naming over the course of 6 months. Before any of you say it, I'll just go ahead and say it myself. Because of the nature of how this graph was generated or what it was generated to demonstrate, it's missing one critical piece of information for a presentation, which is I don't have the sham arm shown here. So the sham is completely flat between baseline and 2 months, no improvement whatsoever. And then, we took, when we imaged these individuals, we did find that they first of all had activation often of the right hemisphere in the performance of the language tests that were performed in the scanner, and also that they had shifts in that representation, shifts in which areas of their right hemisphere were activating during these tasks after they had received 2 weeks of stimulation. Importantly, and a sort of harbinger to something I'm going to talk about in a little bit, what we didn't seem to do was have sort of global suppression of right hemisphere activity in those individuals when they receive stimulation. They changes in patterns of activity. Those changes, by the way, I should say, were fairly individual. They varied a lot between persons. But what we didn't do is we didn't sort of globally shut down the right hemisphere during the performance of these language tasks. And we could demonstrate that in addition to naming, individuals had increases in their spontaneous speech production. So what I'm showing here is pre to post TMS improvements in performance on the cookie theft picture naming description task. And you can see that people had improvements in their number of nouns and closed and open class words, various word types, verbs as well. And importantly, what is not the case we weren't causing people to talk for longer. And because that would be a really boring reason to be able to generate more words, right? If they spent more time describing the task. And they didn't speak any faster. It's actually that the time that they spent performing the task was spent on task. In other words, they made fewer errors during the time that they were speaking. All right, so I want to come back to this idea that we performed site finding, that we searched all over, informed by this kind of interhemispheric inhibition model. We were like, oh, well, maybe suppressing the right hemisphere is the way to go. And we had identified all these regions of the right hemisphere that seemed to be activated in the service of language tasks. And what we ended up finding was that although we were agnostic and tried to approach all of these areas in our subjects, for almost every subject, it was the same area that was the most useful area. Just showing here the green dot, a little bit in the blue dot as well. Same basic Brodmann area. Both of them allude to Brodmann's area 45, which is the inferior frontal gyrus in the right hemisphere, which, just by way of reminder, was that very same place where in our imaging analyses, when we asked that pretty simple question, is there a region of the brain that doesn't seem to be homologous with respect to its patterns of activity? It's exactly the same place, right? And so by a couple of converging lines of evidence, we thought that perhaps what we're dealing with here is a kind of noisy node, that maybe other areas of the right hemisphere may have some purchase with respect to language, but that this particular area, it seemed to be the case that modulating it in an inhibitory way across subjects seemed to have some relevance for improving language function. And, time's gone by. As Laurel alluded to in the introduction, I've been around for decades now. So other people have done this and used this basic same approach of inhibition to the pars triangularis on the right and demonstrated positive signal with respect to favoring the use of RTMS, repetitive TMS, compared to sham stimulation. Now, you might say to yourself, okay, that's great, but you haven't really done anything to disambiguate whether or not the right hemisphere might be sort of monolithically deleterious to language performance. In other words, maybe having right hemisphere activity is bad, and you just happen to pick the worst. You found the best worst spot to inhibit to improve performance against a backdrop of brain areas that are recruited in spurious or deleterious ways, right? And fortunately and unfortunately, one of the cases that we saw in our early work, I think, kind of puts the nail in the coffin of the idea that the right hemisphere is monolithic in its inhibitory input of the left hemisphere in individuals with chronic stroke and aphasia. So this is imaging data from one of our subjects who, like most of our subjects, had their best temporary response to inhibitory stimulation of the pars triangularis, that right inferior frontal gyrus. And what I'm just showing you here, the before and after fMRI images, before and after receiving a single session of TMS, and the subtraction is down below. And so you can see that there's a decrease in bold activity over the area where we were stimulating her, where we were applying her treatment for the trial. Unfortunately for this particular patient, like Barlow's patient from 1877, and one of the reasons why I point to Barlow's patient earlier on, she also suffered from a second stroke. In this particular case, she had a second stroke involving the right hemisphere, again, like Barlow's patient. This stroke was subcortical. So I don't know if you can see, depending on your screens, but you can see the stroke in the diffusion weighted images that are the second row of the slide there, second row of brains. And so it's those bright white spots, they're subcortical on the right. Obviously, she has an old, large left hemisphere stroke. And so if you are a interhemispheric inhibition purist, you would say to yourself, oh, well, it's generally bad for cognition to have strokes. I'll admit that. But maybe in this particular case, since the right hemisphere is exerting inhibitory influences on the left hemisphere and its ability to recover via perilesional areas that are engaged in language, maybe this might end up being a good thing. That you're decreasing the transcallosal connections from the right hemisphere to the left hemisphere. All right, well, it turns out that theory was just not borne out at all. So I'm going to blow this up a little bit. So this is her performance. This patient was kind enough to continue with us after she had had that second stroke. And what we found was that no, after that second stroke, she had a modest decline in other cognitive abilities, really quite modest, but she had a remarkable decline in her language abilities, both her naming abilities specifically and a composite language measure as part of what's called the cognitive linguistic quick test, right? Sort of strikingly compared to her other domains of cognition. So the idea here is that even in this patient, where it seemed to be the case that inhibition of one part of this sort of right hemisphere constellation of brain areas involved in language, inhibiting that one spot was useful. A more global or non-specific inhibition of or suppression of right hemisphere activity, well, if anything, seemed to be quite deleterious, and sort of helping us understand that the right hemisphere's role is not monolithic. A lot of what I've said has applied, by the way, to patients with anterior lesions, because our work has historically focused on non-fluent aphasia. And so I just want to make the point that more recent work has suggested that the dynamics of brain activation between more anterior and more posterior lesions may be different. So this is work from Dorothy Sauer's sort of large research group in Germany. It's sort of well known for following the sort of patterns of brain activity in patients with stroke and aphasia over time. And what they demonstrated is that if you look at lesions that are more anterior, that's what's demonstrated in B. If you look at the sort of patterns of activation after those strokes, it turns out that that homologous regions, right, come online pretty acutely and tend to stay active in those individuals, where you don't tend to see that, at least in her recent study, in individuals with more posterior lesions. In case you can't see it from the figure, the activity of the region that's labeled as the homologous brain area in the more posterior lesions depicted in A is just sort of trailing on the bottom of the graph for the entire graph, right? And so it may be the case that there is topographic specificity to some of the things that I've discussed before, since we've been exclusively looking at patients with non-fluent aphasias. So here's an interim summary before I move on to what we've been doing more recently. And to be minimalist about it, we're going to talk about the right hemisphere's role in recovery, and a little bit tongue-in-cheek, I'm going to label this 5 years of work summarized in 4 words. And so what we learned, from our early work with TMS in patients with aphasia was, well, you could think about whether or not the right hemisphere is helping, hurting, both, or doing nothing. But it's that activity is completely epiphenomenal, right? Those are your options. And so, I mean, I'm just going to take epiphenomenal off the table, right? Otherwise, there'd be no point of modulating these areas. There wouldn't, sort of, none of what I've shown you would have been relevant at all. Well, we also know that it doesn't seem to be the case that all activity of the right hemisphere is helpful, right? If that were the case, then it wouldn't stand to reason that focal inhibition of that brain activity would have a beneficial effect, either temporarily or in an enduring manner for those patients. By the same token, however, it doesn't stand to reason, both by historical data and by the data generated in our own laboratory with the patient who had a second stroke, that the role of the right hemisphere is all deleterious, right? There's clear evidence, and those aren't the only pieces of evidence, that that damage to some of these areas of the right hemisphere might hinder language recovery as well. And so we really think that what we're talking about is a more complex network of brain areas that have compensatory roles, but that maybe the compensatory roles of these brain areas are inefficient, or that certain areas of the brain and its engagement with language tasks is inefficient. And that for us provides a sort of promising territory in which to do neuromodulation, right? Focused modulation of brain activity in order to try and optimize recovery. So I'm going to shift gears now and quickly talk about the other workhorse of the laboratory, which is transcranial direct current stimulation. Although it's often mentioned in the same breath as TMS, it's actually technologically pretty different. It involves applying a sub-threshold amount of stimulation to the brain on the order of 1 to 4 millivolts. So milliamps. So it's really quite modest. It's thought that what happens is that over time, you're nudging lots and lots of neurons, millions of neurons, a little closer, a little farther away from their actual potential firing threshold. So the net effect of that is to alter neuronal firing rates. The received wisdom is that anodal and cathodal stimulation have different effects. I won't call them opposite effects. I'll call them different effects, because that is also debated. However, one of the real advantages of this technology is that it is extremely safe. In the entire history of the device, no one suffered a severe adverse effect from getting TDCS, which is great. It's also an order of magnitude cheaper than TMS, which is great. It combines with therapies because you can wear it. And like TMS, repeated application of TDCS can have long-term changes in behavior that we think are related to neuroplastic changes like long-term potentiation. Now, one of the weaknesses, if you're a cognitive neuroscientist, of transcranial direct current stimulation is that conventional stimulation has pretty poor spatial resolution, right? So here's what's called a finite element model, just basically a current flow map of the distribution of current when you place standard electrodes in standard positions on the scalp. And you can see that rather than just applying stimulation under where the electrode is placed, what you get is this just smear of activity that runs between the two electrodes, right? Because the current just wants to get from one electrode to the other and doesn't care what's in between. It doesn't care about your experiment. And so if you're trying to prove something very specific about a very specific region of the brain, that can be a challenge. However, if what you're trying to do is, slides seem to have frozen. Oh, yeah, okay. If what you're trying to do is demonstrate or is modulate the activity of a distributed bilateral network of brain areas, well, then maybe applying conventional TDCS, something that has pretty poor spatial resolution, could hit a whole network of brain areas at once. Well, maybe that's not such a bad idea. And so we took this technology. We had a very similar design in which we brought people in and we stimulated, with the anode on one side, cathode on the other. We were pretty agnostic about the electrode orientation too. So cathode on one side, anode on the other. We tried to pick an optimal montage for each individual, kind of like when we were trying to pick a best site for stimulation for individuals. And after that baseline, we brought them in and did a pretty similar design to what I showed you before with the TMS, a 2-month, in this case, 2-month study where all the persons who were in the sham arm crossed over into the real arm. And this time I have the common sense to include the sham arm in my graph. So what you could see is an improvement in performance on the Western aphasia battery, aphasia quotient, overall measure of aphasia severity in individuals with real TDCS compared to individuals receiving sham. And once again, because this talk is somewhat historical, we're far from the only people who've done this kind of work now. And what you can see, again, a testimony, in my opinion, to the diffuse nature of stimulation, that there have been successful studies that have applied TDCS, this sort of low spatial resolution technology, in a variety of montages across the language system, across both hemispheres, probably hitting much of both hemispheres with most of these studies, right? And so in my opinion, some of the focal arguments with TDCS are a little bit overstated. And it may be the case that we're often just stimulating much of what is the remaining compensatory language network. Again, this is from a meta-analysis that we performed a couple of years ago. You can now demonstrate that across the studies that have done TDCS in patients with post-stroke aphasia, there's a signal favoring TDCS compared to sham. And I'd be remiss if I didn't mention the most recent large study involving TDCS in patients with post-stroke aphasia done by Julius Friedrichsen's group in South Carolina, where they looked at 74 individuals and demonstrated a benefit of anodal stimulation compared to sham stimulation in patients with chronic aphasia. Before I get to what we're looking forward to doing in the future, I also want to mention that we've started to apply TDCS in other populations with aphasia, in particular persons with primary progressive aphasia, family of neurodegenerative aphasias. And while time doesn't permit me to go through a sort of a description of each of these aphasia subtypes, you could, there are three, and what I'll broadly say is that they divide into a non-fluent agrammatic variety, a variety that has difficulty with semantic processing, also referred to as semantic variant PPA, and a third variety called logopenic progressive aphasia. These individuals have sort of word pauses, problems with pretty dramatic problems with repetition. They're often conceptualized as having a deficit of verbal verbal working memory. The patterns of atrophy associated with these different aphasias is shown in the figure above. Again, because of the limits of time, I don't have time to go through all the pathologies, but we'll talk about that some other time if you like. And in our early work, we were pretty agnostic because PPAs are not all that common, right? So we were pretty agnostic with respect to where we put our electrodes and who we wanted to stimulate amongst these three subtypes. Our goal was to try and hit as much of the peri-sylvian territory as possible, as much of the left hemisphere as possible, so that we can include individuals from all these subtypes and hope that they were getting neuromodulation. And so in our open label pilot work, we brought people in, applied TDCS for 10 days, and then followed them up out to 12 weeks. And we had great results with our open label work. I mean, we could demonstrate across a battery of tests that we made composite measures of our various measures into measures of speech production, repetition, grammatical comprehension, semantic processing, that there were benefits in the individuals who got TDCS. As is often the case, things became, I think, a little bit less bombastic and effusive when we introduced a sham arm, but we were still able to demonstrate that compared to individuals who got sham stimulation, there were a number of benefits in the individuals who got real stimulation, and enough to motivate us to, as I'll talk about in a little bit, pursue larger trials having to do with this technology. One thing that we found as well in our earliest work was that individuals who are more severely affected at baseline seemed to demonstrate a better effect from TDCS compared to individuals who were high functioning with respect to their language performance when they received TDCS. And while I recognize fully that one could make a strong argument that that might be to some extent regression to the mean, I'll point out that in contrast to lots of other work in aphasia where the most severe patients are not great responders to the intervention, it was encouraging to us that our patients who were coming in with pretty severe deficits were our best responders, or at least were good responders to our intervention. And now I would be remiss if I didn't mention another big group that's implementing this work excellently. This is Kirana Tsapkini's group down at Hopkins. And this is from what at the time was the largest trial that had been done in TDCS patients with PPA, featuring 36 patients, in which they demonstrated that individuals with both logopenic variant and non-fluent agrammatic variant appeared to improve as a function of having received stimulation. Not so much the folks with semantic variant. Okay, so that was a whirlwind tour of about, I don't know, 10, maybe between 10 and 15 years worth of work. So I want to talk a little bit about where we are and what's next. And so I'm going to break that out into insights that we've achieved, what our knowledge gaps are, and what our next steps are. And so I hope I've shown you numerous encouraging proof of concept studies. But so far, we still don't have sufficient clinical data to actually use these technologies in a widespread way in clinical environments. So obviously, the next step for that is larger randomized controlled trials. Sorry, it's a little slow today. The slide advancer. Okay, we've talked about interhemispheric inhibition as a kind of useful initial framework for thinking about what's going on with neuromodulation, but the models that guide neuromodulation, that one in my mind especially, are oversimplifications at best. So we need novel tools and models to characterize language system plasticity. I showed you that specific approaches like 1 Hertz repetitive TMS to the pars triangularis seems to be effective, at least for some people. But we don't know if other sites or other parameters are useful as well, or maybe even more useful. So we need novel mechanistically informed sites and stimulation parameters. And then finally, individual responses to neuromodulation vary widely, but we lack the evidence regarding what factors predict that response to neuromodulation. So we need to explore predictive patient features and intermediate markers of plasticity. So I want to show you, I'm going to spend the last 10, 15 minutes talking about what we're doing in each of these domains. So for larger and for more and larger trials, the answer is pretty straightforward. We're running larger and more trials. So we have an ongoing trial, a phase 2 clinical trial of TMS for post-stroke aphasia. Branch Coslet in our lab is the PI for that. And so this is individuals who are getting that same stimulation in the right pars triangularis as we ended up giving in our early work, paired this time with a kind of speech language therapy, modified constraint-induced language therapy. We have 2 ongoing trials involving the use of TDCS in patients with PPA. Those 2 trials include one that employs conventional TDCS, so large electrode TDCS, and then another that employs high definition TDCS, so TDCS that tries to be more focal with respect to its spatial resolution to try and target a specific region. In this particular study, the inferior frontal gyrus on the left. This isn't our work. I just want to show it because I think it's encouraging. What this is an article from last year demonstrating the temporary benefits in a population of patients with chronic fluent aphasia of stimulating over Wernicke's area. So on the left hemisphere in a region that we don't stimulate, in enhancing comprehension performance in this patient population. It was a pretty preliminary study, so it just looked at the immediate post-stimulation effects. But in the vein of looking for more parameters and looking for more ways to stimulate, I think it's really encouraging that that we're able to pursue, say, excitatory stimulation of the injured hemisphere in ways that seem to have salutary effects. I talked a little bit about the need to identify factors that predict response, and I think one of those factors that we need to look at is baseline impairments. This is work that we did. I did with Denise Harvey, who's a former T32 fellow at Moss and Penn, and now in our laboratory, looking at what kinds of errors in our patients with chronic aphasia, what kinds of errors were most predictive of their ability to respond to neuromodulation. And it turned out that if you looked at phonological naming impairments versus semantic naming impairments, that it was their phonological naming impairments that first of all correlated with their overall accuracy in our cohort, but also was the thing that seemed to respond when individuals were responding in this particular study to a single session of transcranial magnetic stimulation applied again to that right hemisphere spot that we're so accustomed to applying it to. I also mentioned in the family of baseline predictors and baseline factors. I mentioned this already once, but in a variety of our studies using TDCS, both in PPA and in patients with post-stroke aphasia, we found that baseline severity was a useful predictor of response to stimulation. We're also interested in differences in cortical plasticity as a predictor of response to neuromodulation. And in this line of work, one of the things that we've been using as a tool is a different form, like a different paradigm of brain stimulation as a kind of index of plasticity. So let me say a word about that. So I've already alluded to this, but you can use a pulse of stimulation over the motor cortex to elicit a motor response, okay? So that's great. That's called a motor evoked potential. Now what you can do is you can take another form of TMS and modulate, apply it to the motor cortex before and after you get these motor evoked potentials. Now we're going to use something called a continuous theta burst stimulation. So imagine now you get a motor evoked potential, you apply this form of stimulation, which is this robust inhibitory form of stimulation, right? And then you start getting more motor evoked potentials. Well, this form of TMS is highly inhibitory, and so what you get is you get this period of time where the cortical response is inhibited. Okay, and so we have been looking at whether or not that period of time or the depth of inhibition can be thought of as a measure of cortical plasticity. Why would I think that's the case? Well, I sort of make the analogy. It's kind of like memory foam, right? If you sort of push, if you perturb the brain, the amount of time it takes for that neurophysiology to bounce back from that perturbation can be thought of as a kind of index of the plasticity of the system. Like, how easy was it to change the system? All right, so if we do that in individuals with chronic stroke and aphasia, we can demonstrate a few things. Well, first of all, we demonstrate some differences in healthy individuals. And one factor that we looked at in healthy individuals to get us on our way was a genetic feature that varies in the healthy population that has effects on brain plasticity. There's this gene, or I should say there's this protein called BDNF, brain-derived neurotrophic factor, and it appears in a couple of different variants in the population, depending on your genotype. Okay, one variant, the evidence suggests, has a sort of weaker effects with respect to plasticity than another. And so what we first did was we demonstrated that you have marked differences in response in terms of this measure of neuroplasticity, motor evoked potentials, based on your BDNF genotype. And so what we showed was, okay, if we did this paradigm again, where we apply motor evoked potentials before and after a theta burst stimulation, and we just stratified people by their BDNF genotype. Either they had Val66 Val. So, both of their genes are Val, or Val66 Met genotype. What you demonstrate is that the individuals with the less efficient, less neuroplastic genotype, they have far less, that's these people, they have far less response to neuromodulation, this inhibitory neuromodulation, than people who have the genotype that's associated with higher neuroplasticity. Okay? And so what we've started doing is we started looking at how this affects our individuals with post-stroke aphasia. And the first thing that we can demonstrate is that in statistical models, we can show that if you add BDNF genotype to models that predict recovery in individuals with chronic aphasia, that you get a significant addition of accuracy, right? Your models become more predictive if you add that genotype. In other words, BDNF genotype seems to matter in terms of aphasia recovery. And then what we most recently found in our reporting on, well now, is that if you then look at the individuals who have that less plastic genotype, that Val66 Met genotype, we actually find that in addition, that response to TMS, which I showed you was so markedly different in those individuals, right? The degree to which they have a sort of normative or non-normative response to TMS in that circumstance is also a predictor of their long-term aphasia recovery, right? It adds information to the model. So again, we're getting increasingly interested in, as we move forward, in factors that affect sort of neuroplasticity and also sort of predictors of neuroplasticity and how that influences aphasia recovery. Finally, in the last 5 minutes, I want to talk about our use of network neuroscience. And this is largely with our partner, John Medaglia at Drexel University. And so for this, I want to start with a sort of obvious truth, which is that cognitive localization in the brain is a function of its local and global connectivity, right? And so from this, we can start thinking about how the connectivity of different spots in the brain, right? How that connectivity dictates what the roles of those areas of the brain are, right? And so, for instance, in this little cartoon model of a brain network, this region of the brain, like that node in the network, clearly does something pretty different or has different properties than that node, right? Which is clearly responsible for connecting or disconnecting those two big modules. And those both clearly do something different than that node, which has one real role, which is to communicate with this node the one node it's connected to. And so we've gotten interested in this idea using something called network control theory, a set of ideas borrowed from neuroengineering that allows for inferences about the operational utility of brain centers based on their anatomic connections. Why are we interested in that? Because we're interested in why those different regions of the brain that I pointed to that either are or aren't helpful for stimulation in patients with aphasia and in healthy individuals, what are the properties that determine or make them useful in that way? And so we don't have time to go into the details of how the network neuroscience was done, but just to boil it down, we looked at the specific control properties. We focused on two, boundary control and modal control, where boundary control is the ability to segregate and integrate sort of large modules. So this area has a lot of boundary control. Modal control is the ability to push networks into very specific states, like this little node that only does one thing. It pushes the network into a specific state because it only interacts with that other node right there, right? And so what we did was we generated maps of these control variables based on the anatomic, not functional, anatomic connectivity of individuals' brains. And what we could do is we could map out individual differences in the distribution and degree of these controllability measures. We had a hypothesis. Our hypothesis is that that in Broca's area, this is in healthy individuals, that there would be differences in performance on different kinds of tasks depending on how much control you had in those areas, and how much you had of these different controllability variables. Specifically, we thought that tasks that were more open-ended, where there were a number of different potential answers that you could generate to the task, would rely more on boundary controllability, or at least that boundary controllability variability would relate to that. Whereas we thought that closed-ended tasks, like read this long number aloud, there's only one way to get a right answer to that. We thought that closed tasks would rely more on modal controllability in areas that are relevant for language. And indeed, we're able to demonstrate, again, in the interest of time, I won't go through all the details, that boundary controllability, if you stimulated individuals in Broca's area, again, healthy individuals, the degree of boundary controllability made a difference in how they performed on open tasks. Whereas if you stimulated individuals and they performed closed tasks, it was the amount of modal controllability that seemed to matter in terms of perturbing their performance on closed tasks. The bottom line being is that we're starting to use individual network anatomy to start to predict responses to TMS. And we're trying to get at some of the sort of deep properties that might help us identify which regions we ought to be stimulating, even in the right hemisphere in patients with post-stroke and aphasia someday. So to conclude, I hope I've convinced you that focal manipulation of the right inferior frontal gyrus with TMS enhances language performance, that diffuse direct current stimulation may facilitate recovery in both post-stroke aphasia and PPA. And, my promissory note for the future, we need bigger studies, we need novel imaging tools, we need better predictors of treatment response. And I think they're on the horizon, in our lab and other labs for neuromodulation in persons with aphasia. I do practically none of this work. I just stand up and talk about it, whereas a lot of other people actually do it. So I just want to give thanks to the various people who worked on this project. This is the current crew. I also want to thank the collaborators that we've had in other institutions over the years, and I'm happy to take a couple minutes of questions.
Thank you, Roy. I think some of us, unfortunately, may need to go to another meeting, and I'm one of them, but I will open it and suggest that those who can stay and want to ask Roy questions or make comments, so please feel free to do so. Thanks.
Looks like everyone's jumping off. Thank you. It was a pleasure speaking to you all today. Bye.